This lecture is on the chapter Mechanisms of Genetic Exchange in Bacteria. So in bacteria, DNA from other sources can enter and can change the genetic properties of the bacteria. There are three basic ways by which DNA can be transferred into bacterial cells. The first is transformation, in which free DNA in the environment can enter into bacterial cells. The second is conjugation, where DNA can be transferred from one bacterial cell into another through the formation of a conjugation tube. And the third is transduction, in which a virus carries bacterial DNA into a host cell and transfers the bacterial DNA into the cell. These processes are demonstrated in the diagrams here. The first shows how transformation takes place. In this process, DNA enters into a bacterial cell. Once the DNA enters into the cell, it aligns with homologous sequence present in the bacterial chromosome and replaces the DNA in the bacterial chromosome through the process of recombination. So in this case, if a DNA fragment with A plus property, that is the property to show a certain phenotypic feature, like let's say the ability to break down a particular substrate, enters into the bacterial cell, which was previously A minus, then A plus can replace A minus through recombination, and the bacteria gains the property of utilizing that particular substrate. That is, it now becomes A plus instead of A minus. The A minus fragment, which is replaced by the A plus fragment, is subsequently broken down by the bacterial host. In conjugation, two bacterial cells join together through the formation of a conjugation tube, and DNA from one bacterial cell can enter into the other. In this case, the bacteria which gives the DNA is called the donor cell, and the bacteria which receives the DNA is called the recipient cell. In the third process, called transduction, bacterial DNA carried by a virus enters into a bacterial host, and if the properties carried by the bacterial DNA in the virus are different from that of the bacterial host, then the properties of the host can actually change through the process of recombination. So from these information, we can summarize that in transformation, cell contact is not required, whereas the process is sensitive to DNase, because if DNase is present in the environment, then DNase breaks down the free DNA molecule in the environment, and subsequently transformation cannot take place. In case of conjugation, cell contact is required because the DNA is transferred from one bacterial cell to another through the conjugation tube. However, this process is not sensitive to DNase because the DNA is present inside the cell or inside the tube and cannot be broken down by the DNase enzyme. In transduction, cell contact is not required because the DNA is transferred from a virus to a bacteria. This process is also not sensitive to DNase because the DNA that is transferred is protected by the virus particle and so is not accessible by the DNase enzyme. So now we are familiar with the three basic processes by which DNA can be transferred from one bacterial cell into another. Let us now look into the processes in a little bit more detail. In transformation, DNA can be transferred from the environment into bacterial cells. This process was first studied by the scientist Frederick Griffith in 1928. He used Streptococcus pneumoniae, the bacterium which is responsible for causing pneumonia in humans. Streptococcus pneumoniae has several serotypes. These serotypes are based on the presence of capsular polysaccharide on the surface of the bacterial cells. Because capsular polysaccharides carry different combinations of sugar molecules, capsules can be of different types in Streptococcus pneumoniae, and as a result, they can be assigned different types, types 1, 2, 3, 4, and so on. These types differ in the composition of the sugar molecules which are present in the capsule. Now, there can be two forms of each type, a smooth form and a rough form. For example, for type 1, some bacterial cells may be able to produce the capsular polysaccharide properly. Hence, they are called smooth because they produce a sugary coating outside their cell on the media, in the media that is, and when they do, they appear as smooth colonies. On the other hand, if they are rough mutants, that is, they carry some kind of mutation in their DNA, which prevents them from making the actual polysaccharide material. As a result, they appear as rough colonies and we call them type R. So for each serotype, that is for type 1 or for type 2 or for type 3 and so on, there can be again two subtypes, type 1R or type 1S, where type 1R is the rough variant of the type, that is, it cannot produce capsular polysaccharide, and the other is type 1S, which can produce capsular polysaccharide. In case of type R cells, they contain the genetic information in them, but because there is some kind of change in that sequence, they cannot produce the capsular polysaccharide properly. So they appear as rough colonies. Another difference between smooth and rough types is that the smooth type is virulent because capsule is a potential virulence factor of bacterial cells. It helps the bacterial cells to fight against the immune system. It prevents phagocytosis and also acts against the complement system. So when capsule is present, the bacterial cells are able to cause infection more efficiently, hence they are virulent. And type R cells, as they lack capsular polysaccharide, they cannot establish an infection properly, and hence they are avirulent, that is, they are not virulent. In Griffith's experiment, Griffith used different types of Streptococcus pneumoniae. The table summarizes the different types of Streptococcus pneumoniae used by Griffith in his experiment. So there can be type 2R cells, which are rough cells that cannot cause disease, then type 2S cells, which are smooth cells that can cause disease. Then again, there can be type 3R, which cannot cause disease and are rough, and type 3S, which can cause disease and are smooth. In Griffith's experiment, he injected mice with type 3S Streptococcus pneumoniae. So as you already know that if the bacterial type is smooth, then it can cause disease. So when mice are injected with living type 3S that contain intact capsule in them, they are able to cause disease in the mice and the mice die as a result. And when the blood sample is collected from the mice, type 3S bacterial cells can be recovered. If the type 3S bacterial suspension is heat killed, that is, if high temperature is employed to destroy the bacterial cells and the suspension is injected into mice, mice remains alive because the cells are now dead and cannot cause disease anymore. To cause disease, the cells not only require virulence factors like capsular material to work against the immune system, but they also need to express other factors which can actually establish the disease process. So just having capsular material is not enough to cause disease. Expression of the DNA is also important to express toxins and other factors which can lead to the disease process itself. So when the cells are heat killed, they cannot express their DNA. They cannot produce the toxic factors that they need to cause disease. And as a result, the mice is unaffected. When heat-killed type 3S cells are mixed with live 2R cells, then you would expect that the mice would remain alive because type 2R, even if they're living, they cannot cause any infection inside the mouse because they lack their capsular material. They cannot fight against the immune system, and hence they are destroyed by the immune system of the mice and the bacteria die. They cannot cause any disease. But when these are mixed with heat kill type 3S, we would expect that mice would still remain alive because neither heat kill type 3S, which are dead, nor living type 2R, which are avirulent, can cause disease in mice. And so we would expect the mice to remain alive. But it was observed that mice died even when this mixture was injected into their body. So the conclusion from Griffith's experiment at that time was that there was some kind of transforming principle which was transferred from one bacteria to another, which ultimately led to the mice dying. But because the type 3S cells were dead and the type 2R cells were alive, it was anticipated that something was transferred from the dead cells to the living cells, because in order to express any activity, cells need to be alive. So it was assumed that something was transferred from the type 3S into the type 2R cells, so that the type 2R cells gained some kind of genetic information that converted them from rough form into smooth form. It was believed at that time that whatever was transferred changed the property of the type 2R, but it was not known what was actually transferred. Some scientists argued that the living type 2R cells might have mutated back to type 2S cells, which produced proper capsular material and hence caused disease in mice. But when bacterial cells were recovered from the blood of mice, they were found to be type 3S and not type 2S. So if type 2R cells underwent mutation, one would expect them to revert to type 2S and not type 3S. This indicated that some kind of genetic information transferred from type 3S into type 2R cells, which made them virulent. Later on, scientists used combinations of different enzymes, including DNAs, RNAs, and proteases, to determine what was affecting this transfer process and hence what the nature of the transferred molecule could be. So basically what they did was, in a tube, they took heat-killed type 3S, leaving type 2R, and in that tube, they added DNAs only. In a second tube, they took the same bacterial combination, added RNAs, and in a third tube, they took the same bacterial combination and added protease. It was seen that only in the tube where DNAs was added, this process was affected and the death of mice did not occur, which meant that the transfer of material from type 3S to type 2R did not take place. It was then understood that the molecule which was transferred between the type 3S to the type 2R was DNA and not RNA or protein. So in summary, we can say that transformation is a process in which DNA is transferred from the environment into a bacterial cell. In the case of the heat-killed type 3S cells, because they were killed, they lysed, they released their DNA, and from that DNA, a fragment entered into the type 2S cells and entered into the host chromosome through the process of recombination. So transformation can then actually transfer information from the environment into bacterial cells. And if the DNA is integrated into the host cell, then the property of the host cell can change. The process of transformation is sensitive to DNAs because DNAs can break down the free DNA in the environment and can stop the process of transformation. And the process of transformation does not require contact between two bacterial cells because the transfer happens between free DNA and the bacterial cell which lives in the environment. The fate of the DNA which enters into the cell determines the fate of the bacterial cell, because if the DNA which enters into the cell finds a homologous sequence, then only it will become integrated into the bacterial chromosome. If it does not have a homologous sequence, then it cannot enter into the cell by recombination, in which case it is broken down by the cell and the bacterial cell remains the same in terms of its previous properties. If the incoming DNA has homologous sequence to the host cell chromosome, Then the incoming DNA replaces the homologous DNA in the host, and the host then has a different property from what it had previously. So that's all you need to know from transformation. The next process is conjugation, in which DNA can be transferred from one bacterial cell into another by the formation of a conjugation bridge or a conjugation tube between two bacterial cells. This process was first studied with the help of bacteria containing F factor. F factor stands for fertility factor because it was found that this factor was needed for conjugation to be possible. That is, for gene transfer to take place from one bacterial cell to another, conjugation needed the formation of a conjugation tube, which was encoded by the F factor. In terms of the presence of F factor, bacteria can be classified into three types. One is F minus, where no F factor is present. Second is F plus, where F factor is present as a plasmid. That is, it is present separately from the bacterial chromosome. And third is when the F factor is integrated into the bacterial chromosome. We then call the cell a high frequency of recombination cell. Why? Because when F factor is integrated into the bacterial chromosome, then the bacteria shows a very high frequency of transferring its F factor to other cells by conjugation when compared to just F plus cells. That is, bacteria with F plus cells free in the cytoplasm have a lesser tendency to transfer the F factor by conjugation than those in which the F factor is present as an integrated form in the bacterial chromosome. So F factor can be present in two states. One is the autonomous state, where the F factor is present free in the cytoplasm, and the other is the integrated state, when F factor is present as a part of the bacterial chromosome integrated into it. Now, what leads to F factor integrating into the bacterial chromosome? So you know that for recombination to take place, homologous sequences will have to be present so that any incoming DNA can integrate into the bacterial chromosome. Likewise, for F factor to remain integrated into the bacterial chromosome, homologous sequences should be present. So in this case, F factor has sequences which are homologous to sequences in the host bacterial chromosome. And because these sequences are very short in size, the process of site-specific recombination takes place. And because of this recombination, F factor becomes integrated into the bacterial cell. When F factor becomes integrated, we call it a HFR cell as opposed to F plus cell, where the F factor is present free in the cytoplasm. Now, when a F factor carrying bacterial cell comes in contact with the F minus cell, then the F plus cell sends out F pilus, which contact the F minus recipient cells. And as the F factor or F pili they contract, the recipient cell is brought closer to the F plus donor cell. When the recipient cell comes close enough to the donor cell, a conjugation bridge forms, and through this bridge, F factor can be transferred from the donor cell into the recipient cell. However, if the donor cell gave away its F factor to the recipient cell, it would itself become a recipient and would not be able to transfer DNA to other cells in future. In order to stop that from happening, donor cells replicate their F factor before the F factor is transferred into the recipient cell. This occurs by rolling circle replication because F factor is a circular DNA molecule. It can be copied by rolling circle replication into two copies. The strand of DNA which is displaced from the F factor during rolling circle replication is transferred into the recipient cell. And as the DNA strand is transferred, Okazaki fragments are formed in the recipient cell, and eventually the whole displaced DNA strand is copied in the recipient cell through the formation of Okazaki fragments. In the donor cell, as the strand is displaced, a 3' hydroxyl end forms at the other end where the displacement takes place. And from this 3' hydroxyl end, elongation of DNA can take place using the inner DNA strand as a template. And when the DNA strand is elongated using the inner strand as template, double-stranded DNA can be regenerated. So in one bacterial cell, the DNA is copied continuously from the 3' hydroxyl end where displacement starts from. And in the other bacterial cell, the strand which enters into the cell is copied through Okazaki fragments. So where the F plus cell is, F plus plasmid is nicked, two ends are generated. One is the 5' phosphate end and the other is the 3' hydroxyl end. The 5' phosphate end enters into the recipient cell, and the 3' hydroxyl end is elongated in the donor cell. So you end up with two copies of double-stranded DNA in each cell, one through continuous elongation in the donor and the other through Okazaki fragments in the recipient. Now, this is what happens in bacterial cells, but we as humans can use this process to our advantage. Since we know that HFR cells have a high frequency of recombination, then we can use HFR cells for the mapping of bacterial DNA, and we can then find out which genes are adjacent to each other in the bacterial chromosome. So a very simple way of carrying this out is by using two cells, one HFR and the other F-, in which DNA is transferred from the HFR cell into the recipient cell. And during this process, since the incoming DNA carries the gene for a certain phenotype, the properties of the recipient cell changes. So based on observing which property has changed, we can determine which gene has been transferred into the recipient cell, so that we know that this is the gene which came from the donor cell into the recipient cell. And if another gene follows the previous gene, we can state that these genes were adjacent to each other in the original DNA. That is why they were transferred one after the other during the conjugation process. So if we take a donor cell which has the phenotype threonine plus, leucine plus, sodium azide sensitive, bacteriophage sensitive, lactose plus, galactose plus, then if this donor cell transfers its genes one by one into the recipient cell, we can determine the order of the genes in the donor cell by studying the phenotypic change in the recipient cell. In this case, the genotype of the recipient cell is just the opposite of those in the donor cell. That is, if the donor is threonine plus, the recipient is threonine minus. If the donor is leucine plus, the recipient is leucine minus, and so on. So what do these phenotypes actually mean? How do they help bacteria, and what do they do inside the bacterial cells? So threonine plus basically means the bacteria can synthesize the amino acid threonine. You know that bacterial cells require many amino acids because they use these amino acids to synthesize their proteins. So if certain amino acids are not produced by the bacterial cells, they cannot synthesize their proteins properly, and hence they cannot survive properly in their environment. Threonine plus is an enzyme which synthesizes threonine amino acid and enables bacterial cells to synthesize proteins subsequently. Leucine plus likewise is the gene which allows the synthesis of leucine, which can be used for forming proteins. Sodium azide sensitive gene is the gene that makes a bacterial cell sensitive to the bacterial inhibitory agent sodium azide. So sodium azide is a chemical which can be used to inhibit bacterial cells or to destroy bacterial cells. Certain bacterial cells, they possess the sodium azide resistant gene, in which case the cells are not affected by sodium azide and they remain alive, whereas others carry the sodium azide sensitive gene, that is the AZIS gene, which makes them sensitive to that particular inhibitory agent. TON-S stands for bacteriophage sensitivity gene. That is, if the bacteriophages are present in the medium, bacterial cells are killed if they contain TON-S gene, whereas they are resistant if they carry TON-R gene. LAC+ and GAL+ are genes which enable bacteria to use lactose and galactose as sources of energy and carbon. That is, if you do not provide them with glucose, but you provide them with either lactose or galactose, they can still break down lactose and galactose as their sources of energy and can survive subsequently. Now, the most important is the STRS gene. This is the streptomycin sensitive gene. This streptomycin sensitive gene is what differentiates the donor cells from the recipient cells. Donor cells are streptomycin sensitive, whereas recipient cells in this case are streptomycin resistant. So whenever we carry out any gene mapping study by conjugation, we have to use a marker which can differentiate between the donor cell and the recipient cell. In this particular case, since the transfer of properties in recipient cells is observed, we need to have recipient cells which are resistant to a particular antibiotic, so that after the end of the transfer process, if we plate the bacterial cells in the presence of this antibiotic, then only recipient cells will survive and donor cells will die. This will allow us to remove all possible donor cells and study only recipient cells. And this is what we want, because we want to see which gene has been transferred into the recipient cell. And so we want only recipient cells to be present in the bacterial culture. Another important factor is we have to make sure that the streptomycin resistance gene is not present close to the area that we are trying to map. Because if it is present close to the rest of the gene cluster, then it is also likely to be transferred between the donor and the recipient. So if that happens, we cannot differentiate between donor and recipient anymore, and the gene mapping experiment may fail to work successfully. So we have to ensure that the antibiotic resistance gene is far away from the part of DNA that we're trying to map. Now, the way we carry out this experiment is, first we mix the HFR cell with the recipient cells, and we incubate them at a suitable temperature for a short period of time, because conjugation does not take place over a long length of time. So let's say about 5 to 7 minutes of initial incubation is a good option. So when we mix them for 5 to 7 minutes, if one cell is HFR and the other is F minus, then the HFR cell expresses F plus, pulls the recipient or F minus cell closer to it, and starts transferring its F factor into the recipient cell. Now, in the case of F factor, there are two origins which are present. One is ORI-T, which is the origin of transfer, and the other is ORI-V, which is the origin of replication. There is also a third origin which is present in F factor, that is ORI-S, which is a secondary origin of replication. The function of ORI-T is to act as an origin of transfer of the F factor from the donor cell into the recipient cell. So whenever transfer occurs during conjugation, it starts from ORI-T. ORI-V is the origin of replication of the F factor, which is used for replicating the effector inside the bacterial cell. ORES is a secondary origin of replication, which carries out replication of effector only if OREV becomes non-functional for some reason, like, for example, mutation or some kind of change in the sequence of OREV during the lifetime of the bacterial cell. So when conjugation takes place in an HFR cell, transfer starts from the ORE-T of the effector. And ORE-T of the effector can be present anywhere along the length of that integrated effector itself. So as transfer starts from a random position within the effector of the HFR cell, then along with effector, some adjacent DNA sequences of the bacteria can also be transferred, because if ORE-T is not present at the end of the integrated factor, and if it is present along an approximate or random position of the effector, then when that is transferred, the whole of the effector may have been transferred, that is starting from ORE-T to where it ends in the integration site. All of that sequence can be transferred, and beyond that, certain bacterial genes may also be transferred. So if we allow conjugation to take place for about 5 to 7 minutes or 10 minutes, then within that time, ORE-T transfer will start. From ORE-T up to the point of integration, effector transfer will occur. And if we allow conjugation process to continue, then 1 or 2 genes from the bacterial cell will also be transferred. So what we do is we allow conjugation to take place for some time. And after conjugation has taken place for some time, we vortex the cells, so that as we vortex the cells separate from each other and conjugation stops. Because you know that vortexing puts a lot of pressure on the bacterial cells, puts a lot of force on the bacterial cells. And because of that force, the cells separate. So when the cells separate, we know, or we assume, that 1 or 2 of the bacterial genes have been transferred along with the part of the effector, which was transferred by conjugation. So to determine which gene or genes have been transferred, we need to make plates for each of those genes which are present in the HFR cell. That is, we need to make 2 plates, one containing threonine, the other without it, one containing leucine and the other without it, a third plate containing sodium azide and another without it, a fourth plate containing bacteriophage and another without it. Then we use one plate where we provide only lactose as the source of energy, and another that contains glucose and lactose as source of energy. We make a 6th plate that contains only galactose as the source of energy, and another containing glucose and galactose as their source of energy. In all these plates, the antibiotic streptomycin will need to be added, because remember, we are trying to study only recipient cells. So if we add streptomycin to all the plates, only recipient cells will grow. Now, since we don't know which gene will be transferred first from the donor to the recipient, we have to use all 6 plates containing the different factors that help you to identify which gene was transferred. Because we don't know, if we knew the map already, then we wouldn't need to go through this experiment. So since we need to know the sequence of genes which were transferred, we have to make all 6 plates, and we have to test the bacteria in all of those 6 plates for every time interval that we allow conjugation to happen for. So after 5 or 7 minutes, if we see that the recipient cell, which was not able to utilize lactose as a source of energy, can now grow in the medium containing only lactose as a source of energy, then we know that since it was not able to excuse me, since it was not able to synthesize lactose as its source of energy, but can now do so, it must have acquired the lactose plus gene from the donor. If the recipient cells are not able to grow in other plates in which it was not supposed to grow, like recipient was threonine minus, so you would expect it to grow only in threonine plus media. If it is still not able to grow in the absence of threonine, then you know that it did not get the threonine gene from the donor. Same for leucine. Recipient was leucine minus. So after this conjugation phase, if it cannot grow in the absence of leucine, that is in the plate in which you did not add leucine, recipient did not grow. The plate in which you added leucine, recipient grew, meaning that recipient is still not able to synthesize leucine. That is why it is only growing in the plate when leucine was provided. Then you would know that recipient did not receive the leucine gene or the threonine gene from the donor. In case of sodium azide sensitivity, recipient was resistant to sodium azide. So if it did not get the sodium azide gene from the donor, it would still be sodium azide resistant and it would still be able to grow in the presence of sodium azide. Same for bacteriophage resistance. Recipient was already bacteriophage resistant. So if it can grow in the presence of bacteriophage in the plate where bacteriophage was added, then you will know that donor did not transfer bacteriophage sensitivity gene to recipient yet. In case of lactose and galactose, if recipient can grow in the absence of glucose, that is, if it can grow in the presence of lactose, or if it can grow in the presence of galactose, then you know that recipient received the lac plus gene from the donor or the gal plus gene from the donor. That is why it is now able to grow in the presence of lactose or galactose as the only source of carbon and energy. So based on your observation, you have to interpret which gene was transferred. Is the recipient now growing in the absence of threonine? If yes, it got the threonine plus gene. Is it growing in the absence of leucine? If yes, it got the leucine gene. Is the recipient now sodium azide sensitive? Then it got the sodium azide sensitivity gene. Is it sensitive to the bacteriophage? Then it got the bacteriophage sensitive gene. If it can utilize lactose as the only source of energy, it got the lac plus gene. If it can utilize galactose as the only source of energy, it received the gal plus gene from the donor. So based on your observation, you have to interpret which gene was transferred first. If only one gene was transferred after 5 to 7 minutes of incubation, then you know that that gene was immediately adjacent to the F factor integration site. So you would place that gene first in your gene map. If two genes were transferred during these 5 to 7 minutes of conjugation, then you have to repeat the experiment using a shorter time duration so that only one gene would be able to transfer from the HFR cell into the recipient cell. So in this way, by carrying out the conjugation process for different lengths of time, you can determine which gene was transferred, and you can also determine the sequence in which the genes were transferred. So by looking at the time of transfer, you can plot a map of the genes that were transferred, and you will get something like this, where you got the sodium azide sensitivity gene transferred after 8 minutes of conjugation, the bacteriophage sensitivity gene being transferred after 10 minutes of conjugation, the lactose utilization gene transferred after 18 minutes of conjugation, and the galactose utilization gene transferred after 25 minutes of conjugation. So in this way, you can determine the order of the genes in the original bacterial chromosome, and you can then plot a map which you can show in this form, where you use origin of transfer as your reference point. So you place that at 0 time, because that is when the F factor started getting transferred. After 9 minutes, sodium azide was transferred. After 11 minutes, bacteriophage sensitivity gene was transferred. After 18 minutes, lactose utilization gene was transferred. And after 25 minutes, galactose utilization gene was transferred. You can also show threonine plus and leucine plus genes in the map if you know the time of transfer which allowed these genes to be transferred, and you can then also show them in the map. Notice that the two sensitivity genes were transferred one after the other. That is, sodium azide sensitivity and bacteriophage sensitivity were transferred one after the other. The lac plus utilization and gal plus utilization were also transferred one after the other. So this makes you realize that genes with similar properties usually occur adjacent to each other. That is, sensitivity to sodium azide and sensitivity to bacteriophage, two properties which affect the survival of bacterial cells in the environment, they usually occur adjacent to each other. This means that when bacterial cells transfer genetic information between themselves by conjugation, they usually transfer those genes which are likely to be needed for the survival of the bacteria in the environment. For example, if the environment contains various inhibitory factors like sodium azide or bacteriophage, then it would help the bacteria to receive those genes together. That is why the sodium azide sensitivity genes and the bacteriophage sensitivity gene came adjacent to each other during conjugation. Same for lactose plus and galactose plus. Since both of these are sugars which are likely to be present in the environment, the genes for their utilization also come together. So if the bacteria finds lactose in the environment, it will break down lactose. If not, if it finds galactose in the environment, it will break down galactose. So this helps the bacterial cell to survive better in the environment. If it can get genes responsible for sugar breakdown together, then it becomes more competent to break down different varieties of sugar. If it gets genes for sensitivity together, like sodium azide sensitivity or bacteriophage sensitivity, then it can fight in the environment with other bacteria, along with other bacteria, I meant, against sodium azide and bacteriophage together. So these are similar genes which are transferred at the same time, and as they have similar properties, they help bacteria in a similar way. The next process is transduction. And in transduction, bacterial DNA is transferred from a virus, that is through a virus, into a bacterial cell. But before that, I would like to mention a little bit about sexduction, which is done by F prime factors. So you know that any sexual process involves the transfer of genetic information from one cell into another. And in case of HFR cell, when the F factor is getting transferred into the recipient cell, then some bacterial DNA is getting transferred. But when the bacterial DNA is getting transferred, then the incoming DNA can replace the DNA in the recipient cell, and the bacterial cell does not become a diploid cell. So if the F factor is in the form of a HFR cell, then the HFR cell only sends out a open-end DNA fragment into the recipient cell. And when the open-end DNA fragment enters into the recipient cell, it can replace the host cell sequence by recombination. So the host cell still has only one copy of that sequence, and so the host cell remains haploid. But in case of F prime factor, since F prime factor is a circular DNA, if it enters into the bacterial cell, just like a plasmid does, then the bacterial cell gets two DNA molecules. One is its chromosomal DNA, and the other is the F factor. And since they can exist independently inside the bacterial cell can be called a partial diploid because the F factor only carries some genes of the bacterial DNA, but not the whole bacterial chromosome. So we can call the recipient cell as a partial diploid. In case of partial diploids, sometimes because there is significant homology between the DNA sequence which is present in the F factor and in the DNA sequence which is present in the bacterial cell, recombination can take place and the bacteria retains only one of the genetic information. It destroys the other genetic information. If that happens, then the bacteria becomes a haploid again. So when the F factor enters into the bacterial cell, the bacteria may remain as a partial diploid or may subsequently become a haploid if recombination happens between the F factor and the bacterial chromosome. So when F factor becomes integrated into the bacterial chromosome, then its sequence remains as a separate sequence in the host chromosome. In this diagram, you can see the F factor sequence as a purple sequence and the bacterial sequence as a blue sequence. Now, when F factor detaches from the bacterial chromosome, we use the term excises from the bacterial chromosome. Then sometimes what happens is the F factor, as it entered into the bacterial cell by recombination between homologous sequence, it is supposed to excise from the bacterial DNA once again through recombination between those homologous sequence. That is what is expected. But sometimes what happens is F factor can accidentally carry out homologous recombination with a sequence which is not the original homologous sequence that was used for integration. So integration happened between a sequence of the F factor and a sequence of the bacterial chromosome which were homologous. So when excision happens, that is the opposite of integration, you would expect the same sequences to come side by side, and you would expect the same sequences to cause recombination and release the F factor from the bacterial DNA. But if the F factor finds a different sequence in the bacterial DNA, which is homologous to it, then it might carry out recombination with that sequence. And this is very easy for F factor to do because site-specific recombination takes place in the case of F factor, in which very short DNA sequences are involved. And if very short DNA sequences are involved in site-specific recombination, then those short sequences are likely to be present in different places along the length of the bacterial chromosome. And because of this, F factor may find a short stretch of DNA in the bacterial chromosome to which its ends are homologous and can carry out recombination with those sequences instead of the original sequences used for integration. So when that happens, F factor, because it makes a mistake, recombination causes some bacterial genes to be present in the F factor, whereas some parts of the F factor may be missing, or the whole F factor may be there, but because of mistake in the excision process, some bacterial genes may also be there. Such a factor, which carries F factor along with some bacterial sequences, is known as a F prime factor. So F prime factor contains F factor plus bacterial sequences. When this F prime factor enters into a bacterial chromosome, then that bacterial chromosome also has similar sequences that are carried by the F factor, F prime factor. And because of this, the bacteria now has some sequences which are present in duplicate. And when this happens, we say that the bacteria is a partial diploid or a merodiploid. So such a merodiploid bacteria or partial diploid bacteria can continue to survive in this form. That is, the F factor stays independently, the host chromosome stays independently, in which case the bacteria continues to be a partial diploid, or in rare cases, the sequence of the F prime factor, which are derived from the previous bacterial host, they can replace the sequences in the bacterial host chromosome, and the bacteria then becomes a haploid cell again. It does not remain a diploid cell anymore. So it depends on the fate of the sequences. If they remain in the F prime factor, the bacteria is diploid, whereas if they replace the host sequences, then the bacteria becomes a haploid cell. So in this case, we call the bacterial cell, if recombination takes place, then we call the bacterial cell recombinants because they appeared or they occurred as a result of recombination. So we call them recombinants. That is, they arose because of recombination between the F prime factor and the host cell. So moving on to transduction, which is the last process of gene transfer from one bacteria to another. Remember, we said that transduction is a process in which bacterial DNA are carried by viruses. And when viruses infect new hosts, then they transfer the bacterial DNA into the host. And so the bacteria gets DNA from a different bacteria with the help of that virus. So in a normal viral infection of a bacterial cell, two processes can occur. One is where the viral DNA enters into the bacterial cell. The viral DNA takes control of the bacterial cell. It produces many copies of the viral DNA, many copies of the viral head and tail, and eventually it lyses the bacterial cell after it has formed complete virus particles using those DNAs that it replicated, those virus heads and tails that it produced inside the host cell, using all the resources of the host cell. This is known as the lytic cycle because the bacterial host cell lyses after infection by the virus. Another is the lysogenic cycle in which the virus DNA can integrate into the bacterial DNA. It can remain integrated in the bacterial DNA for several generations of the bacteria, during which the bacteria replicates the viral DNA along with the bacterial DNA. It's only when the bacteria faces some kind of adverse condition in the environment, such as the depletion of nutrients or the exposure to UV, is the viral DNA excised from the bacterial DNA so that the virus can form new virus particles and can infect bacterial cells in other environments. Since the current environment is not suitable anymore, the bacteria progresses to infect cells in other environments where those environments are suitable for the bacterial cells to survive and hence suitable for the viral DNA or the virus particle to be propagated. Now, transduction can be either generalized transduction or specialized transduction. In generalized transduction, the virus packages random copies of the bacterial DNA instead of viral DNA. So the virus head contains only bacterial DNA and no viral DNA. This is because when the virus tries to make copies of its DNA, the bacterial DNA also breaks down into smaller pieces. And when the virus packages the DNA into its head, it does not recognize that the DNA belongs to the bacterial cell and not to the virus itself, and accidentally packages the bacterial DNA into its head. When this virus subsequently infects a bacterial cell, then it injects the bacterial DNA into that cell. So the bacteria then gets a copy of those sequences in its cell, even though it already contained those sequences in its chromosome. So the bacteria can either replace those existing sequences with the viral DNA, or the viral DNA can become integrated in a different part of the bacterial DNA. So if the viral DNA replaces the bacterial DNA, then the bacteria becomes haploid. If the viral DNA integrates into the bacterial DNA at a different location, then the bacteria becomes a partial diploid. In other cases, the virus can carry its own DNA along with some bacterial DNA. And this occurs only when the virus DNA integrates into the bacterial cell. And when it excises, once again, it confuses some other sequence during excision, and it carries some bacterial DNA along with it during the excision process, so that when it subsequently makes virus particles, it carries those bacterial sequences along with the virus DNA and introduces those bacterial sequences into the next host when it infects that host along with its own DNA. So when bacterial DNA only is carried by the virus, the process is called generalized transduction. And when bacteria plus virus DNA is carried by the virus, then the process is called specialized transduction. So for specialized transduction, the lambda virus is a very common example of a virus which can cause specialized transduction of E. coli. The lambda viral DNA can become integrated into the bacterial DNA when the virus infects the E. coli host cell. When the environment in the of the bacterial cell is suitable, that is, it is conducive for the growth of the bacterial cell, then the lambda DNA can become integrated into the bacterial cell DNA and carry out a lysogenic cycle as opposed to a lytic cycle. During this lysogenic cycle, if the environment of the bacteria changes so that the environment is not suitable for the bacterial cell survival anymore, then the viral DNA detaches from the bacterial cell. Now, once again, when the lambda DNA detaches, it can make mistakes by carrying out recombination, homologous recombination with a random bacterial sequence, which is present close to the site of the integration of the lambda DNA. So it can accidentally carry bacterial DNA which are present adjacent to the two ends of the lambda DNA. That is, if the lambda DNA integrates into the bacterial cell, and if it integrates between the biotin gene, which synthesizes the vitamin biotin, and the galactose gene, which enables the bacteria to utilize galactose, then if it makes a mistake, depending on which end made the mistake, the viral DNA can carry either the galactose gene or the biotin gene, depending on which end did the recombination process, and as a result, which type of bacterial gene was carried. So if the virus DNA carries galactose gene, we call the virus lambda D gal, that is lambda virus, but defective with the galactose gene. That is, it contains the galactose gene, which is a defect in the lambda virus because it is not supposed to carry that gene, or lambda D bio virus, that is lambda virus, but defective, does not carry the full lambda virus sequence. It carries the biotin gene, which is its defect because it is not supposed to carry the biotin gene of the host. So the fates of the lambda D gal or the lambda D bio, they depend on which gene is missed by the virus during this imperfect excision process. That is, when the virus makes a mistake, did it accidentally leave its lytic cycle gene or did it accidentally leave its lysogenic cycle gene? If when the virus detaches from the bacterial cell, if it leaves some of its lytic cycle genes, but still carries the integration genes, then it can enter into a cell and it can integrate its DNA into the bacterial chromosome. But because the virus lacks lytic cycle genes, it will need to remain integrated into the bacterial chromosome forever. Because if it detaches or excises, it cannot carry out the lytic cycle because it is missing the lytic cycle genes. If lytic cycle genes are missing, then the virus cannot carry out the lytic cycle. It cannot produce copies of itself through the lytic cycle. So it will need to remain integrated into the bacterial DNA forever. However, if the bacteria is infected by a helper virus, that is a virus which carries all the genes intact, then that helper virus can help these defective virus without the lytic cycle genes to multiply and to leave the cell through the lytic cycle. So basically what is happening here is the helper virus produces everything it needs, but because it produces everything in the cytoplasm, the defective virus has full access to those things that are needed, and as a result, it uses those resources to produce copies of its own DNA by the lytic cycle. The helper virus makes its particles at the same time. The helper virus also carries out lytic cycle, but at the same time, the defective virus can also produce particles of itself because the helper virus has produced all the enzymes and other proteins that it could not produce because it lacked some of its genes. On the other hand, if the virus carries the gene for the lytic cycle only, but not the genes for the lysogenic cycle, then it can only carry out the lytic cycle and produce copies of itself. It cannot integrate and it cannot carry out the lysogenic cycle of the virus. So it depends on which gene is missing, what the virus is going to do, what kind of cycle the virus is going to pursue, whether it will go for the lytic cycle or it will go for the lysogenic cycle. So remember that as with any other incoming foreign DNA, the fate of the lambda D gal and the lambda D bio also depends on what happens to that gal gene. If the lambda virus DNA remains intact, separate from the bacterial DNA, then the bacteria becomes a partial diploid. Whereas if the lambda, the galactose gene or the biotin gene in the lambda DNA, if they replace the host bacterial DNA, then the bacteria does not become diploid, it becomes a haploid again. So, it can either be a cell with 2 copies of the gene, or it can carry only 1 copy of the gene. But the replacement of the host DNA by the virus DNA is a rare process. So usually as a general rule, bacteria remains partial diploids. We can use this concept to infect bacterial cells with viruses carrying different genes to see how the presence of those genes affect the nature of the bacteria. Like, let's say we have a bacterial cell and we think that the one fragment of the galactose utilizing protein may have a more important effect on galactose breakdown than another fragment. So in this case, what we can do is we can incorporate the fragment for the gene for one of those fragments in the lambda virus, and we can use that lambda virus to infect a host cell to see if that particular fragment is present, then does that affect the activity of the host bacterial cell. So let's say we know that for lactose utilization, bacteria needs 2 different peptides in a protein. Okay, so for lactose breakdown, bacteria needs an enzyme, and that enzyme has 2 different types of peptides present in it. And we want to investigate how those 2 peptides differ in their function. Is one peptide more important than the other, or are both peptides needed for that particular function? So in order to carry that out, we can create a bacterial cell where one of the genes for the 2 peptides is mutated so that only one peptide is produced. We can study the behavior of that peptide in that bacterial cell. We can then introduce the mutated peptide gene by using a lambda virus, which carries the gene as a defective gene. That is, it is a lambda D, that gene which you're studying. We can use that virus to infect the bacterial cell and see that if the virus carries the correct form of the gene in its DNA, and if the bacteria then becomes a partial diploid where its own chromosome lacks that protein fragment, whereas the viral DNA carries the bacterial fragment, does that make any difference or not? So if mutating the gene for one fragment makes a difference in the bacterial cell, and if then subsequently providing the bacterial cell with that same fragment in the right form makes a significant change in terms of breaking down the substrate, then we know that that fragment plays an important role in the breakdown of the substrate. Otherwise, that fragment is probably needed for the structure of the enzyme, and it might just play a physical role in the enzyme. So you can carry out different genetic experiments. You don't have to know in detail about this experiment, but this has an application where you want to study the role of a particular DNA sequence or a particular gene in bacteria. So you just mutate that gene in the bacterial cell, and then you supply the gene through a virus to see if that makes a difference. In this case, if you use only lambda defective virus, then sometimes you don't get enough bacteria which have been transduced by the virus, because like any other gene transfer process, the transduction also occurs at a very low frequency. Usually on a general basis, 1 in 10 to the power 6 cells, that is 1 in a million cells, undergo this kind of gene transfer. So if you use only the defective form of the virus, then you don't get enough transductants. So it becomes difficult to study the effect of that gene. So what you can do is you can use a 50-50 ratio of the two forms, that is the wild type lambda virus and the defective lambda virus, in equal proportion. And you can use that mixture to infect bacterial cells. And like we said before, since the defective viruses, they miss some important genetic information, if you provide the normal virus at the same time, then the normal virus can produce the missing factors to the defective virus, and the defective virus can then propagate, and at the same time, it will transfer the bacterial DNA into the cell too. So if you want to make your experiment successful, the transduction experiment successful, then you can use a combination of the wild type virus and the defective virus, in which we call this high frequency of transduction, that is HFT. And after transduction, if the bacterial cell undergoes lysis, then you get high frequency transduction lysates when the two different types are mixed together, as opposed to if you use only the lambda defective virus, in which case you get low frequency of transduction, or LFT. And you get low frequency of transduction lysates, that is when the bacterial cell lysis after the lytic cycle of the virus, you will get only very few copies of the bacteria that actually carry the virus DNA in it. So you can either go for LFT, where you use only the defective virus, or you can go for HFT, where you use a mixture of the lambda virus with the defective virus, so that you get many copies of bacterial cells with the viral DNA present in it. So that brings us to the end of this chapter. In this chapter, we learned about 3 different processes of gene transfer, namely transformation, conjugation, and transduction. And we also learned the different experiments that were carried out in the olden days to study the different processes. And also you learned about the consequences of gene transfer in bacteria through this video. So I hope that made the chapter clear to you, and it helped you to understand how different gene transfer processes occur in bacteria and how we can use them to our advantage. For example, we can use conjugation for gene mapping. We can use transduction for studying the function of different genes or DNA sequences in bacteria through the process of transduction. And we can also use transformation to our advantage, where we can transfer antibiotic resistance gene or a substrate utilization gene into a bacterial cell by using free DNA from the outside of the bacterial cell. So I hope the chapter was clear to you, and that's all for now. I wish you all the best.